In this video, we will discuss about the high trap that are usually found in the colorectal cancers can cause chemo-resistance and immune suppressions. And later on, we will discuss how the researchers has targeted this TRAP1 to increase the chemotherapy and the immune checkpoint therapy efficiencies. So in the panel A, what they did is they checked the effect of these genes on the tumor cells and in the normal cells. And what they are able to find out that in the case of tumor cells, uh, these genes are upregulated compared to the normal cells, and out of this they are able to see that this TRAP1 genes that are highly upregulated in the tumor cells compared to the normal cells, and they check it for the coed cells and the reed cells. That is shown in the right hand side of the panel A and B. For the reed cells is the bottom panel, and for the coed cells it's the top panels. And then they check the survival rate for the low TRAP1 and the high TRAP1, and they are able to see that high TRAP1's patients has high survival rate compared to the low TRAP1s. And they also check the TRAP1s using the Western blot and compare it with the normal healthy cells, and they can see that in the tumor cells TRAP1s are higher, and the relative mRNA levels also shows that TRAP1 mRNAs are higher in the tumors compared to the normal. The panel D shows the immunofluorescence image, where we can see that in the case of tumors we can see the more red signals, which is which is the signals for the TRAP1 compared to the normal cells. And the panel E is the immunofluorescence image of the single cells, where we can see that TRAP1s can be seen clearly. And another one is the control vectors, and the third one, first one is the controls, where we can see that it's the TRAP1 and there is no green signals are there. And second one is these vectors, where we can see the green signals, and the third one, in this case we do not see any TRAP1, or we can say that TRAP1 has been knocked down. There is the guide RNAs for the TRAP1, in which case TRAP1 has been knocked down, and is the guide RNA for the control, where these green colors is being shown. So this is the system that they used to knock down TRAP1s. And there is the another image where they shows that cytochrome C uh, cytochrome C translocations got affected in the case of where the TRAP1 is knocked down or not. The hypothesis is that the TRAP1 forms a complex with the cytochrome C and prevents the translocation of cytochrome C uh, into the cytosols or outside the mitochondria, and thereby, since the cytochrome C does not comes in the mitochondria, outside the mitochondria, it does not triggers the this it does not triggers the this apoptosis mechanisms in the tumor cells. So here, when they knock down this TRAP1, we can see that the cytochrome C seems to be comes out of the uh, membranes, or can be find in the comes out of the mitochondrial membrane and can be found in the cytosols. Whereas in the case of this control, where TRAP1 is not knocked down, we do not see the presence of the cytochrome C. And these things they have shown in this panel G also, where we can see that uh, it shows the Pearson coefficients, where the cytochrome C is it shows the cytochrome, it shows the SGRA, SG TRAP1, SG vector, and the control or the Pearson coefficients, or we can see that I think it reflects the cytochrome C in the mitochondria. So we can see that in the case of control and SG vector, cytochrome C's in the mitochondria are higher, but in the case of TRAP1, cytochrome C's in the mitochondria are lower. And here, when they check the Western blot for the cytochrome C within the cytosols, we can see that cytochrome C in the cytosols are higher compared to the case for the control and SG vector. Next, you check the markers, that is, uh, these are the immunosuppressant markers GR1 and FOXP3, and this is the immuno activation markers, immune activation markers CD4 and CD8. So in the case of control, where TRAP1 is not knocked down, you can see the activation markers are lower, but suppression markers are higher. But when the TRAP1 is knocked down, we can see that these activation markers are higher and the suppression markers are lower. So this all indicates that when the TRAP1 has been knocked down, then it can triggers the apoptosis of the cells, and that can also helps in the immune activations and reductions in the immune suppressions. And these things they did studies on the colorectal cancers. So brief overview is shown in this panel J, where we can see the colorectal cancers. The researchers developed a CRISPR-Cas9 oral injection systems that can penetrate the mucus, and once it is able to penetrate the mucus, it can enters inside those cells, epithelial cells. It can enter inside the epithelial cells and also in the tumor cells, where it can form a guide RNAs against the TRAP1 and can suppress this TRAP1 formations, and thereby these cells, tumor cells, become more uh, acceptables by the this immune activations. These tumor cells now become more susceptible to be attacked by the immune systems, and this is how the inside of the cells works. So when this TRAP1 is not been knocked down, so in that case the TRAP1 and cytochrome C forms a complex, and once they forms a complex, the cytochrome C is not been able to come out of the mitochondria, and thereby it does not triggers the apoptosis or reactive oxygen species related signals. But when this Cas9 with that can target as TRAP1 is introduced, then there will be no TRAP1, or less of TRAP1 formations will be reduced, and this will cause the cytochrome C to move into the cytosols outside the membrane, and it will increase the ROS, and this ROS will cause the or further accelerate the cytochrome C formation release into the cytosols, and it will trigger pro-apoptotic proteins and downregulate anti-apoptotic proteins, and this will cause this MPT driven necrosis, and this will further activate the immune systems in the tumor microenvironment. Because of this MPT driven necrosis, other parts of the dead cells are more exposed to the immune cells. But when this is closed, when the cytochrome C does not come out of the cells, then what it will does the reverse thing, that is, it will increase anti-apoptotic proteins and suppress pro-apoptotic proteins, and that will cause formations of this FS5FU resistant cells that will maintain this immune suppressive environment in the tumor microenvironments. So this is how, by targeting the TRAP1, they can make the cells more susceptible to the immune system and the chemo resistance, that we will discuss in our subsequent videos. That's all for this video. I hope you understood some of the basic overviews how this TRAP1 will cause chemo resistance and immune suppressions in colorectal cancers. So that's all for this video. Thank you for your time.